I'm really excited to introduce our next speaker, Barbara Englehardt, who just recently moved to the Bay Area to join us, and we'll be talking about the really cool stuff her lab is up to. Thank you for
Yes.
Awesome to be here. I've kind of been moving the last month, so my brain is in boxes pretty much, but it's really great to see everyone here. I'm going to be switching topics to sort of the analysis of this data, the other side of it. So hopefully this is going to get you dreaming about what you can do with your samples after you collect them and normalize them and do everything nice with them. I'll say, backing up just 1 second to sort of talk to the question that you were just talking about, but really quickly. We did a lot of work with GTEx and the flash frozen samples there, looking at principal components and stuff like that of the histology images. The first PC always correlated with ischemic time, 100%. It was absolutely ischemic time. And even more complicated, the genes that were associated with ischemic time were different for every tissue. So I would actually caution against finding those bellwether proteins. I think they'll probably be maybe even different for every tissue. So just as a warning there, I think it's a really hard problem, actually. It's something that I struggled with for about 2 years, like a couple years ago. It's hard. Okay. Well, this is kind of weirdly centered, but here we go. Okay, so I don't need to introduce this to this crowd, but spatial transcriptomics and proteomics are super exciting. Right now, we've gone in my analytic lifetime, we've gone from bulk RNA sequencing. Well, we've really gone from microarrays to bulk RNA sequencing to single cell RNA sequencing, and now we get spatial resolution of single cells in a tissue. It's just super exciting because now we get to understand how cells interact with each other, what goes on inside a cell, what goes on in neighborhoods of cells. It's just super exciting, and I'm really interested in building models for this. In particular, when I think about building these models, I think about, from the statistical standpoint, building spatiotemporal models to be able to understand these tissues. So in particular, what I'm interested in studying is the spatially and the temporally resolved state of cells, what they look like in a snapshot and what the dynamics look like, how they change in space and how they change in time. And in particular, I'm interested in quantifying the intrinsic state of cells, right? What the state of a cell looks like when the variance is not affected by its local environment, and the extrinsic state of cells, right? What the variance of a cell looks like that's affected by its environment, that changes due to what's around it. I'm sort of quoting there Aviv Regev. She talks about that a lot, and I really appreciate the word she uses there. Very importantly, I think I want to understand the healthy and unhealthy interactions between cells. So to do this, I'm sort of trying to develop 4 different types of models, and you're going to see examples actually of all 4 of these today in the 2 examples of methods I'm going to talk to you about. So the first thing I'm interested in doing is studying the spatially resolved patterns in cell state and tissue organization, right? I just want to model cell state, right? And model what tissues look like. The next thing I'm interested in doing is looking at variability in cell state across location, across samples, across populations. We're going to get into that a little bit less, but you're going to see that we're going to be able to look at variability, how state changes, how specific genes change as you wander across a tissue, in this case, proteins in there. I'm interested in predicting the effects of specific interventions on spatially resolved cell states. So say you want to go in to a particular space in a tumor, inject, for example, CAR-T modified immune cells, and see what the effect on those tumor cells are. I'd like to be able to use my model to predict whether those CAR-T cells will effectively kill those tumor cells or not, using the prediction models that they have. And I think, excitingly, design targeted interventions for these perturbations to cell state. In other words, I want to design experiments using machine learning. I want to be able to predict which experiments are going to lead to the results that we want. So the first model that I want to tell you about is de novo alignment of spatial samples. So hopefully when you are thinking about the data that you're collecting through maybe technologies, you're going to have a handful, let's say, slices of brain or a specific tumor or liver, let's say. And one thing you might want to do is take those samples and align them. You might have different marker sets. You might have different resolutions. You might have different machines or different ages of the machines, etc. So we're really interested in thinking about how to do that. Because in order to be able to actually think about variation, you'd like to be able to have a similar coordinate system. Let me say this slightly differently. So spatial samples, in order to be able to understand variation, require an identical coordinate system. Unaligned samples, when you analyze them separately, you can't get variation. You don't know at a specific location really anything about variation, right? But when you are able to align them, all of a sudden, you're able to understand the variation that occurs at a single spot, right? You're able to figure out that at a specific XYZ location, there's actually some level of variation because you have alternative samples that you can project onto that location and you understand about error, right? Does that kind of make sense? So thinking about alignment in order to get variation, we'd like to understand variation in expression and tissue morphology, the variation in interactions and correlation, and variation in cell type heterogeneity. I think that's something very interesting. Something that comes up a lot is how much variation there is said the other way, how much generalizability there is in tumors. And I think this is a subject that we can talk about in a lot of different ways. It very much depends on the tumor and the tumor type, but there is, I believe, some type of generalizability in various tumors, and that's one thing we're starting to look for in specific tumor types. So there's obviously, again, as you very well know, different types of distortion in spatial data. There's both technical distortion and biological distortion. The technical distortion might come from tissue preparation, handling, and sequencing. The biological distortion might come from biological variation, right? What we're interested in doing here is essentially controlling for as much technical distortion as we possibly can by aligning the samples to a common coordinate system in order to be able to essentially deconvolve or dissect the biological distortion, right? We'd like to take apart the biological distortion to understand where each of the different parts of the variation come from. Maybe which part of the variation here comes from disease, comes from sex, comes from age, comes from genotype. And we'd like to remove the technical distortion so as to be able to deconvolve that biological distortion, okay? So that's our goal with this alignment. So this is kind of a complicated slide, but it gives you a really high level overview of what we're doing here. So we have our images here, our bunch of slices, let's say, of a single sample. We're going to warp them all using these weird looking warps. They are locally nonlinear, as you can see. They look like little furry gradients or something like that, but you can see that they're nonlinear, right? I mean, you can see that they're kind of in many different directions, in different locations, but locally they're fairly smooth, right? So that's one property of them. So we warp every single location, every single cell, and we warp each of those cells to a common coordinate system. The cool part about this, I think, is that common coordinate system is unknown. So this is a reference-free alignment. So we can actually start out with 3 or 5 or 100 different samples and no known coordinate system. We can align these samples to a reference-free coordinate system. We can essentially build a coordinate system from scratch, and then we can build this F function, which basically takes every point in this coordinate system, let's say it's a 3-dimensional coordinate system, and identifies for every marker that we have the distribution at that XYZ location of that marker. So for a particular marker then, if we wanted to query in that coordinate system an XYZ location, we'd get a full distribution of what that marker would look like. And we can do that for any marker. So that's a full imputation across our coordinate system of any marker, okay? So that's kind of a nice thing. So what we're calling this thing is an atlas, right? What we're calling this coordinate system here, I don't know if this thing works. What we're calling this coordinate system right here, plus the representation of the markers at each location is an atlas. Because again, in this representation, we can query any three-dimensional point in this coordinate system for any marker that we have access to, okay? And get a distribution. Okay, so here's actually some examples. This is just simulated data with a single gene or protein, however you want to think about it. We have the X or the crosses and the dots. You can see they're observed, and then the aligned, you can see the nice sort of warping there. It looks like a blanket that's been jiggled a little bit. Here's an example from spatial transcriptomics data from 2016. These are 4 parallel slices of breast cancer biopsy. You can see that they actually cover slightly different fields of view, slightly different total coverage of the sample, but we're able to align them in 2 different ways. This is actually 2 different views of our alignment. Here's the observed data space X, right? This is the actual original data X aligned in the original coordinate space. And here's the reference space. This is our the coordinate system that we actually estimate in the second, in the right-hand panel. And this is what I'm calling our atlas, because each of these locations there has the distribution for a single gene in this space. Actually, I don't know what this gene is. I should have written it down, but I don't know what the gene that we actually described there is. So we actually took these exact same 4 tissues. We know that they were parallel slices of the same breast cancer tumor, and we imagined a z-axis too, because we know that they were parallel slices, and it actually worked just fine. So now we have a 3-dimensional alignment too. So here actually is the warping. This doesn't work. So here's actually the warping x and z-axis, and here's the warping if we imagine a y and a z-axis. So you can see the 4 different slices, and you can see the warping for each of the 2-dimensional projections of the different 4 slices. So we have now what I would consider a 3-dimensional atlas of a breast cancer tumor. And again, because of the way that we've described this in our model, we can actually query any 3-dimensional location, any XYZ coordinate within this space, and get the expression values for any gene within this marker space, okay? With any gene within this ST data set that we have, okay? Okay, so that's the first thing I wanted to tell you about de novo atlas construction from a set of samples. The second thing I wanted to tell you is essentially a way to annotate it. So now we have an atlas that we've constructed from alignment of various samples. The next thing we can do is annotate it. And the way we're going to annotate it is by using what we're calling spatial dimension reduction. So the way that we're doing that is with this model. I'll be honest, this model is not necessarily what I wanted to show you right now. The math is not as exciting as it should be. This paper was actually just accepted in Nature Methods. I'm super excited. It's sort of a joke in my lab that I've submitted like 18 papers to Nature Methods, and this is the first one that's ever been accepted. Yeah, I'm really old. Okay. Okay, so spatial dimension reduction basically takes these spatial images and does dimension reduction on them. It's very easy, but it does it in a spatially aware way. Sorry, I'll really quickly tell you what's going on. So you have weights here. These are just going to be our loadings, if you know about dimension reduction, and you have a set of factors. And the idea is that every single cell is going to have a set of factors, right? I'm going to show you pictures of the factors later. And the weights are just going to be weights representing the genes that are representing or make up each of the factors, right? And the idea is that when we do spatially aware dimension reduction, we're basically going to ask every single cell that is nearby each other to end up with the exact same factor values. Okay, that's all we're doing. We want very correlated factor values for cells that are nearby each other in space. That's all we want to do. Okay? We could hypothetically do the same thing for genes or proteins that are similar to each other, in some sort of network, but we didn't do that. We're just doing this with cells that are nearby each other in XYZ coordinate space. And the way that we enforce that is by this Gaussian process here. Okay, and I should say we messed around a lot with how we did that. As you know, sometimes there are pretty sharp borders in tissues, right? Sometimes there are cells that are actually quite nearby each other that shouldn't actually be in the same region, like in brains, and I'm going to show you some example of that. So we actually ended up using a kernel that allows for pretty sharp transitions. So it's not the smoothest kernel on the market, if you will. So here's some examples of what we found. So this is slide CP2 data. This is from the Stickles et al. Paper. This has, as you probably know, about 36,000 observations, 36,000 cells. It really is single cell resolution. A very nice high resolution, fairly small field of view. And this is, mass hippocampus, I should say. So this is what the non-negative spatial factorization looks like. So let me sort of orient you to what this is. This is all non-negative, I should say. We are assuming that the data that we're given are counts. And we could talk about this in terms of the MIDI data. It would be awesome if we could get non-negative data to be able to do this, because we can build a really cool parts-based analysis for you. It sounds like we can do that. So that's really cool. So this is, again, all counts-based or non-negative data. It doesn't actually matter if it's not a count. But this is all a count-based data set from gene expression. This is 10 spatial factors, spatially aware factors that we found. And what I'm doing here is coloring the factors themselves. These are the values of each cell in the factor, in the E to the F, or it's actually just the F that I showed you before in that model. And what you see is they beautifully identify these really precise cell type specific regions of the brain. In fact, my absolute favorite one here is the number 10, the meninges, which is an incredibly thin layer of cells that separates two different brain regions. We saw that and kind of flipped out because we had absolutely no idea that we were going to actually see the meninges in this, but we do. 8 is also a really elegant one too. The dentate gyrase granule layer, I think, is just really well highlighted there. Another thing I want to get at here, which is really cool, is that a lot of these factors really nicely pick out specific cell types, right? But the cell types are overlapping. So some of these very nicely pick out astrocytes or neurons, but those neurons are different in these different regions. In other words, the astrocytes in the dentate gyrase granule layer look very different than the neurons in the cerebral cortex. I'm making that up. I actually don't know if we're doing those two there, but you can go back and look in the paper and see exactly which cells are highlighted there. And you can look at the top genes in each of the different regions, and they're very, very different, which I think is super interesting because again, the same cell types look very different depending on which region they're sitting in, and you can see that very easily in this. So here's actually, interestingly, the top genes highlighted for each of those 10 factors that I just showed you. And they're very different than the factors themselves, right? So the factors look very, very clear and crisp, and the genes look pretty diffuse. And the thing I want to show you with this is that I think sometimes marker genes actually are poor proxy for regions generally. Because in this case, the top gene, and actually it turns out the top sort of 20 or 30 genes, do a pretty bad job of pulling out the same regions that these larger factors do. Here are two examples of non-spatial components, and again, interestingly, they pull out the exact same types of cells. You know, we found astrocytes and neurons, but these are sort of the diffuse, non-region specific astrocytes and neurons that are kind of everywhere, that aren't specific to a particular brain region. And yeah, these are, the argument is that you can kind of see a little bit of spatial organization in 5, but really I think what you're seeing is not spatial organization, you're seeing just sort of a lack of presence of those astrocytes in that particular region of the brain. I would call these very diffuse. So yeah, so those are what the non-spatial factors look like. So we actually, what I'm trying to say is we ran this with non-spatial factors too, and we see very, very different results. Okay, so here's another example with non-negative spatial factorization applied to, this is 10x Visium data. So these are not single cells, these are averages over multiple cells now for every sort of site, every XY, or every XY coordinate, sorry, and much larger fields of view here. So this is what it looks like now, slightly different, but also very clear and crisp regions of the mouse brain again. So the first one's actually fairly diffuse. But the second one, you very clearly see the cerebral cortex, and then we get to the basal ganglia. The fifth one is this nice olfactory granule layer. And again here, although I haven't labeled them, you can actually pick out pretty nicely sets and subsets of genes and cell types that are highlighted within the factors. And again, here are the top genes for each of the different factors. And again, you can see that it's very diffuse instead of the sort of very crisp factors themselves. So one thing we can do here, which I think is kind of exciting, but this is a first pass and not the right thing yet, but we're trying to make this more precise, is actually quantify spatial versus non-spatial variation. So we actually ran this model with both spatial and non-spatial factors together. We wanted to pull out variation that is spatial and non-spatial at the same time and see how much we got. So we can actually just normalize that, right? We can figure out how much variation is spatial, how much variation is non-spatial. And again, this is not right. This is not precise in any sense, but as just sort of a back of the envelope kind of calculation, we can actually try and figure out what's going on with each of the genes that's on the left-hand side and each of the spots or cells on the left-hand side. And you can see that there's actually quite a distribution of how much spatial variation there is and how much non-spatial variation there is. So I think that's kind of interesting, and I think there's a long way to go with that, but as a first pass, it's kind of a fun thing to try. So because of this audience, I wanted to show you this, the results of this Gaussian version. So this is not the exact same model, but it's the Gaussian version of it. So this is, we're assuming the values go from negative infinity to positive infinity instead of counts. For imaging mass cytometry data. So this only has 35 markers, so it's more along the lines of what you guys are looking at. Single cell resolution, and these are fairly small tumor cores, but again, I think it's more along the lines of what you guys look at with the MIDI data. So we applied the spatial factorization, and this is what we found. I really like these. These aren't as crisp as the ones that you guys saw before, because this is a Gaussian version, but I suspect that with the MIDI data, when we do the non-negative version, I think it's going to be much crisper. But we still see a lot of really cool things, and in particular, I really love the fourth one here, where we see really clean vascularization in the tumor cells. And then when we look at the non-spatial representation, again, you see very diffuse, but often very clear cell types that are sort of diffused everywhere, including CK tumor type cells in the second one, which I think is very nice. Okay, I think actually that's all I had to say. That ended quite abruptly, but I'd love to. I did this last night. I'd love to work with you in your data and build some atlases and annotate those atlases. So actually backing up really quickly with this factorization stuff, we can run this on the atlases that we have. So we haven't done that yet, but we can do three-dimensional versions of that, run it on the atlases and annotate those atlases with the factors that we get. So all of that is absolutely on the table, and we've actually proposed that to do that with a bunch of different data sets, and we'd really be excited to work with you all and your data. Thanks.
Okay, I have a question.
Yeah.
We have done something kind of broadly similar with this spatial LDA
Cool.
approach,
Yeah.
which is, developed with Calico, where they basically add a spatial prior onto the latent layer allocation to try and get at a similar aspect. One of the things that's been really nice, and it sounds like this would also work for your approach, is that because it's not a binary assignment, because there's weights of each cell to each factor, you can make like a heat map basically and like show the transition. Is that the case that with either of these approaches, you have the underlying probabilities and can kind of see the transition zones and all that?
Exactly. Yeah, that's right. There's no single assignment. So LDA and this model are both, sort of hard to say it appropriately, but what I would call sort of admixture models, right? Where you can be a part of every factor if you wanted to, right? Or every component if you wanted to. So absolutely, yeah. So that's right. There's no binary, there's no like in or out assignment. That's right. Yeah.
And what about using instead of, so our data is non-negative, it's gonna, the counts of everything is gonna go from, 0 and up.
That's awesome.
The other thing we played around with though, is using cell assignment as the input rather than the marker expression.
Great.
Have you guys tried that or how do you think that would work? Because the reason we do that is because a lot of hand curated time goes into figuring out what the true cell type is. And I think sometimes it does a better job than the actual expression. And then you have basically like a one hot that you could feed in and then use that to try and understand what the neighborhood is.
That's a really good question. So as I kind of hinted at, I think cell types are actually kind of hard to use here. I hear what you're saying. I'm going to throw out some other possibility, which is, so actually what we've done recently, this is kind of a little bit in the weeds, but we've actually developed something called a multi-group Gaussian process, which takes not only like a spatial location, like an XYZ coordinate, which you have already, but a categorical variable as well. So I actually have a rotation student who just joined my group officially, working on adding that to this exact model. And the idea is to not just regularize by location of the cell, but regularize by location plus cell type assignment. Both location and cell type assignment will squish you into the same factors. Does that make sense?
Yeah.
Yeah, so that's on the way. I absolutely agree with you though. I think that is going to be a game changer here. Yeah.
Awesome.
So actually, and I think we chatted about this briefly, like when we met with this smaller group a few months ago. But can you skip? You're showing the spatial influence. Those graphs. So something, and I think today we're going to be for everybody else listening, and we'll be going into kind of the strategies we've used for some of this, which are good, but I think are, especially for like the way we try to assess. And so a question we are always trying to address is you have different cell types, and what is the best way to identify when a cell type expresses a functional marker, that expression of that marker is driven by its spatial location and proximity to other cells.
Yeah.
And and the ways that we've gone about doing that, have been, I think, have been like sort of like pairwise distance estimates and against null distribution. But this strikes me as like perhaps a next gen way to kind of do this in perhaps a little bit more elegant fashion. And so I was going to ask, is that, you think this is adaptable to that, in terms of like thinking in terms of like, if you wanted to look at how does functional marker expression in cell type see what is the spatial importance of, is that?
Absolutely. So we've been calling this the spatial importance for exactly that reason, right? So I think it's exactly that. So, I mean, I think you need to go back and look very carefully at what those factors look like and, sort of convince yourself that they are spatial or non-spatial, right? Which we kind of did, but I don't know if the reviewers were 100% convinced. Because, I mean, I think it's because, that looks vaguely spatial, right? I don't think it is, but, it looks vaguely spatial. So, I guess what I'm trying to say is I think we do need to clean it up and define what spatial versus not spatial means, right? But I agree with you that I think this is the next gen version of that, where we can actually model it and describe sort of the uncertainty associated with those scores, which is, we can't do that with this. So yeah, I would be excited to sort of roll this out and make it more precise. Yeah.
And then the follow-up question I had on that was that, the other thing that we've talked a lot about is that oftentimes you're trying to identify sort of cells that tend to be located relative close to one another, or like we're talking about markers, functional markers seem to be upregulated when two cells are close to one another.
Right.
But identifying ones that are significant, even at against a null distribution that accounts for like canonical tissue structure that you don't care about,
Yeah.
if that makes sense.
Yeah. Absolutely.
And so it seems that with this, could you use like some of these different factors as priors?
That's right. Absolutely. Yeah, that's right. So you could consider this, right, sort of the healthy background, whatever you want to call it. Yeah, that's right. And then control for that as sort of normal, null, whatever you want to call it. That's right. I think that's a great way to do it, actually. That's really smart. Yep.
Okay, question from the chat. Do you have an idea of why there's such a big discrepancy between the marker genes in each cell type and the actual spatial factors?
Yeah, I mean, I think it just takes a lot of marker genes to identify some of these regions. I think that's sort of fundamentally it. I think also it takes a lot of non-marker genes to identify them too, if that means anything. So it takes a lot of zeros as well, right? If that makes any sense. So we need to know what genes aren't there and what genes are there, in this case, genes or proteins as well, to be able to identify these regions. Yeah, I mean, again, the paper has all of or I think it has the top 10 marker genes, but really you need sort of more like 50 to be able to get the colors this beautiful. It's a much deeper bench than I was expecting. Yeah, it sort of made me rethink marker genes, especially, I mean, this is obviously 2 brain examples, so it's very specific to the brain, but it sort of made me rethink marker genes specific to brain cell types at this point. Yeah.
Barbara, when you guys were playing around with optimizing this, did it clean it up at all if you did something like a PCA transformation beforehand and used eigenvectors like people would for like a UMAP embedding instead of working with the raw gene values?
Yeah, good question. No, absolutely not. Great, good. Yeah. So actually this is specifically, in my opinion, so clean because this is working with the count data. So it would be cool to be able to do that, to get a bunch of basically independent whitened, this is what happens when you do the PCA, right? Whitened gene values, eigengenes is what they sort of refer to it as. But actually that doesn't work because those values are on the reals now, right? They're not they're negative and positive, they're not counts anymore. So the reason that this is, in my mind, so beautiful is because when you do non-negative matrix factorization, like LDA is actually, you get this beautiful parts-based analysis, which basically tears apart the littlest things. So if you go to like the original NMF, non-negative matrix factorization paper in Science in like 2000, there's this beautiful faces example where if you run PCA, you just see all of these like really creepy ghost faces, right? But if you look at non-negative matrix factorization on the same data set, you see like an eyebrow and the bottom of a nose and like a lip. And it really pulls out the individual parts. And the reason is purely geometry, right? If you do everything non-negative, then you can only add things to the face. You can't take anything away, right? So this is the same idea, right? Where you can only add things to these sets of factors to make up the full sample, right? So here you're basically saying, I can only add the meninges. I can only add, the dentate gyrate granule layer. I can't take anything away. So I'm not going to put a whole bunch of stuff in each factor. I'm going to put one little thing, right? And so that's why I really like the parts-based analysis thing, the parts-based analysis representation. That's why we actually never went, we kept the counts. And that's why I'm really excited to play with the MIDI counts. That's going to be really cool. Yeah. That's a long answer to your question.
Yeah, Lorenz.
I know the question is towards the atlases from the first part of the presentation. So not atlases from like development biology where like they look at model organisms and they are super stereotypical, and then you can map like whatever sample you have onto the C elegans from the atlas, and then impute, for example. Cancer is like super not stereotypical, right? And like my question is that like what can you do with it? Like when there is an atlas, like a three-dimensional spatial transcriptomics atlas, what do people do with it?
Yeah, I mean,
the question for you.
oh yeah, sorry, for Zoom. Yeah, so the question is with the first example, so here's, you know, sort of a cartoon of the model. If there is an atlas, what do you do with this? And if there isn't an atlas, like, how do you even think about this?
Especially for cancer. Like,
What's that?
especially for cancer.
Especially for cancers, yeah, if it doesn't generalize. So the first one actually is really easy. It's a bizarre answer, but you can actually just sort of plug in the atlas where the common coordinate system is. So right now that's in the model it's what we call a latent variable, right? But actually it doesn't need to be. We can hard code it as that atlas. So we're doing it right now with the Makosco group, with the Allen brain atlas. They have like 112 parallel slices of a mouse brain they've aligned to the Allen mouse brain atlas. And we're just plugging in the mouse brain atlas right there and aligning it. So that works fine actually. So you can do that with a model that we have and just get the alignment. That actually works fine. The other question though is I think the sort of more profound one. And I think it's harder. I genuinely don't know the answer. So to sort of answer the question in a backhanded way. So in this case, we just aligned 4 samples from a single tumor, right? So the atlas is just, you know, the reference points of a single tumor. But really the question is, you know, if we aligned 4 different tumors, is there any common coordinate framework shared across those tumors, right? That I don't know. And I genuinely would be worried about. I think this model could absolutely fail if there were no common coordinate framework across those 4 different tumors. Yeah.
Would you know that it failed?
Yeah, you know that it failed really quick. Yeah.
Well, that's fine.
It
Like,
would probably,
It's only a problem when you don't know that it failed.
Oh, yeah.
It's fine.
Yeah, that's fair. No, it would probably just like align the samples like to totally different regions of the space. That's what would probably happen. Yeah.
Another question from here, is it fair to interpret the non-spatial components as one of the factors, but a factor that doesn't vary spatially?
Let me think.
Wait, what? Can you get that again?
When you separated the spatial components and the non-spatial components. Could you have the non-spatial component just be one of it's like a spatial factor, but it happens not to actually be spatial? Or is it somehow separate from the actual spatial factors that you?
I would think about them very separately, right? I would think about them as factors that, where there's no requirement that the cells have any relationship based on location, right? So again, if we're going to look at pictures of this, yeah, I'm trying to think about it. Yeah, in here you can see like there's no requirement that the color values here for each of the cells are related at all by space. So I don't know if I would think about it. I mean, I would think about it just as sort of a factor free for all, right? As opposed to like, you need some organization, right? As opposed to these where you're saying, oh yeah, you know, this cell and this cell are probably going to be very well correlated across all of these factors simply because they're right next to each other, right? Yeah, that's, I don't know. That's my best interpretation.
Awesome. Thank you so much.
Thank you.